Hi, today we're going to take a look at a G protein coupled receptor pathway. First, let's take a look at the overall process in which this occurs. Now, let's look at each individual step. Okay, first let's talk about what GPCRs do. Essentially, GPCRs monitor things like our growth, our mood, and other aspects like visual and behavioral aspects. Here, the red structure that you see on the screen is our receptor, and the receptor is made up of 7 transmembrane alpha helices. The ligand that binds to the receptor can be anything from a light-sensitive compound to pheromones, hormones, and sometimes even neurotransmitters, for example, dopamine or epinephrine. GPCRs bind to G proteins, hence the name G protein coupled receptors. Before we look at the signal pathway, we should familiarize ourselves with the structure of the G protein. G proteins are proteins that bind to GTP and GDP. In general, G proteins are small proteins that have one subunit, but when referencing GPCRs, the G protein is heterotrimeric, which means they are made up of 3 subunits, the alpha, beta, and gamma. The alpha and gamma units of the protein are attached to the cell membrane via lipid anchors. Right now, the protein is inactive, so the G protein is binded to GDP, or guanosine diphosphate. The reception stage of this signal pathway is a process in which the ligand binds to the receptor, which fits together like a lock and key. Once the ligand binds to the receptor, the protein will undergo something called a conformational change, where the protein changes shape, which then triggers a chain of events that carries out the signal. Once it undergoes the conformational change, the alpha subunit will bind to GTP, or guanosine triphosphate. Now that the GTP is bound to the alpha subunit will disassociate itself from the beta and gamma subunits. Now the alpha subunit will regulate the function of its designated protein. Keep in mind that the beta-gamma dimer can also regulate other proteins and relay messages, but we're going to focus on the alpha subunit. Once the alpha subunit activates a target protein, the protein can then carry out messages via second messengers like calcium ions and cyclic AMP, which will produce a cellular response. As long as the ligand is bound to the receptor, the transduction and response stages can occur as many times. Once the ligand leaves the pathway, it will cease. To deactivate the receptor, GDP will bind to the alpha subunit, which will then reassociate itself with the beta-gamma dimer, and the ligand will then leave. Epinephrine or adrenaline is our fight or flight mechanism. Epinephrine binds to an adrenergic receptor, or basically our GCPR. Once it does that, the adrenergic receptor will undergo a conformational change. The GDP will swap out with GTP and the alpha subunit will regulate its target protein. In this case, the protein it regulates is called adenylate cyclase. When adenylate cyclase is activated, it will change ATP into CAMP, or cyclic adenosine monophosphate, by taking out 2 phosphate molecules from the triphosphate. Once this occurs, the CAMP is our second messenger, and it will then carry out the cellular response such as increased heart rate and the breaking down of glycogen to glucose.